you already introduced me and the talk. So I can only add to what you said, that I'm also part of the GRASS development team, and now I'm currently serving as a PSC chair of the project. I'm a PostGeo charter member and a Phosphor-G enthusiast and advocate. So how will this session be organized? I will show you a short introduction about GRASS GIS, like generalities. Then another basic intro about temporal GRASS, and then we move to a demo session, like using both GRASS on one side, and then we move to R. We connect GRASS to R. Okay. So what GRASS are we talking about? Well, this GRASS stands for Geographic Resources Analysis Support System. It's a free and open source software GIS that we use for geospatial analysis, image processing, graphics and maps, spatial modeling and visualization. And just on July, on 29th of July, we became 38 years old. So it's about like almost 4 decades of continuous development of free and open source software. For more info, you can go to the link in the slides to get like all the timeline of GRASS history. And I also leave you here a link to a video from the 80s, narrated by William Shatner. So it's a little bit of geek GRASS history for those of you that are interested. Now, general stuff about GRASS, as I said before, free and open source, meaning that you can use, modify, improve, share. It has a strong user community and commercial support as well. It is used in different academic, commercial, governmental settings. It has a large amount of tools, more than 500 core modules and more than 300 add-ons or extensions in an official repository, but then different community members or devs also develop their own add-ons and keep them on their personal repos that you can also install from GRASS. We have a graphical user and a command line interface. The software also comes with Python APIs and libraries. And there's just out of the oven new library called GRASS Jupyter that you can try online in this link. So it will open Jupyter Lab and you can just try GRASS online. We also have connection with R, that is like the topic of this session, with QGIS, with WPS, and also with the cloud, using a new package of software called Actinia that uses kind of GRASS in the background. We support different data types like raster, including, of course, satellite images, 3D raster or volumetric raster voxel, vector and space-time data sets that are also like the object of this session. Okay, these modules in GRASS are organized according to what function they have or which input data they take. So in that way, we have like these G dot modules for general data management, R dot modules for raster, and so on. And the same will happen with add-ons, okay? And add-ons are easily installed with a module doing general stuff like G extension here, okay? And as I said before, you can install add-ons from other repos, not only the official GRASS repo, add-ons repo, but other community members repos. So how does GRASS look like when you get to enter the software? You get this graphical user interface. This is for version 7. We have an upcoming version 8 that you can then have a look at the extra slides at the last part of this presentation. But basically, we have a layer manager with different tabs. So you can, as in most GIS, that you handle the turn off, turn on, different layers and so on, and a map display. And the other thing that opens when you start GRASS is a terminal. Okay, so depending if you are in Linux or Mac or Windows, you get like bash or command or whatever Mac uses. And you can control your GRASS session from the terminal. And since the topic of this presentation is how to connect GRASS with R, here is just a quick view, because then we will go deeper in this, on how we connect GRASS with R. So there are two ways. We can either call R within the GRASS terminal. So here we can just type R and R starts within the terminal and it's connected to GRASS. Or we go to either like the command line of R or RStudio, and we use R GRASS 7 library. We load it, and then it's connected to GRASS. Okay. These are two links that you can follow to see examples on how to use each way. Now, having finished with the general introduction, let's move to the temporal support in GRASS, which is what we will be using to process some data and then move to R. So GRASS was the first FOSS GIS that incorporated these capabilities to manage, analyze, and process spatiotemporal data, as well as the relationships between them. We call it this temporal framework within GRASS, we call it TGRASS, and it's basically fully based on metadata, so we do not duplicate any data sets, and it uses a snapshot approach, which means that it just adds timestamps to map, like as extra metadata to map. And then a collection of these timestamps maps is called the space-time data set. Okay, and this will be like then the input or output or for the temporal modules. Then maps within a space-time data set can have different spatial and temporal extents. Okay, but so this is allowed, but for certain operations, you need, as Gilberto was saying before, a data cube. Okay. So space-time data sets can be made of rasters and raster 3D or vectors. Okay, and in GRASS 8, adding another field to the metadata called band reference, GRASS will be supporting image collections as well. So you will be able to register like Sentinel scenes, for example, or Landsat scenes, okay, with all the bands within a unique space-time data set. Again, the modules are organized similarly. So T dot something for general temporal tasks, TRAST for temporal raster data, and so on. And this is just a screenshot with all different group of modules for temporal tasks within GRASS. So you can either see them here in the graphical user interface, or just go to this tab in the layer manager and you get all the modules that you can use. Now, other TGRASS notions that are important to understand what we will be doing. Within GRASS, the time can be defined as either intervals, so with the start and end time, or instances or time points in which we only have like a start time. For example, the timestamp of a Landsat scene or a Sentinel scene is just one point, okay? Then time can be absolute, like in calendar, okay, with dates and hours and minutes and seconds, or relative. For example, when you do simulations, so you only care about which time interval, for example, a map represents, and that's it. It doesn't matter the date. And this is what we can see here, no? Like the time instances, the time intervals. We can see that there are like different time intervals within a time series or gaps and so on. And then the granularity is the greatest common divisor of all the temporal extents in a space-time data set, okay? So for this one, it will be probably the smallest, okay? And the other thing that it's the other concept important within temporal GRASS is the topology. The same way we have topological vectors, we also have this concept of topology among time series and among maps within time series. And there are different topological relations that are considered and are used then in modules, in temporal modules, like equivalent, overlap, during, contains, and so on. And it's represented here with these dark bars. And this is like a general framework and workflow for the temporal GRASS. So you will see just in a moment that all the data that we work with in GRASS must be imported within the software because it has like a native format, okay? And that becomes what we call the GRASS database, okay? But to use the temporal modules, we do not duplicate those data, we just add metadata to the maps, saying, okay, we create a table and we register the maps with metadata in this table. And this is what the temporal database represents here. So basically, with the command tcreate, we create this table, empty, and then with tregister, we add metadata to the maps in the spatial database, and then we complete this table, which is basically the space-time data set, okay? So it's fully based on metadata, sorry. And then we have visualization tools. We have modules to list, to extract parts of those time series, to get statistics for the time series and for the maps within the time series. Tools for pre-processing, like adding colors, filling gaps, setting the null values, for example. And then the core, let's say, of this temporal framework are the aggregation modules like TRAST series, TRAST aggregate, the algebra modules that allow like tons of different operations. They allow you to combine spatial topology, temporal topology, temporal algebra, and different arguments, let's say, both for raster and for vector data. And then we also have modules to import and export time series from GRASS. And there are some other like useful add-ons that can, let's say, complement somehow the core modules for the T-GRASS framework. Okay, we will be using just next some of these modules. Okay, so what will we do? The demo session, now we move to like this third part. In this demo session, we will be doing habitat suitability or species distribution modeling for the Asian tiger mosquito in northern Italy, based on variables that will be derived from daily land surface temperature data. Okay, so within GRASS, we will import the species record, create background points, and create different environmental layers. Okay, this last item using temporal models. Then we will move to R, and there we will read data from GRASS into R, do the model, the species distribution model, the model evaluation, and visualization of results. Okay, so basically, within this workflow, which is pretty similar to what Hannah was showing at the beginning this morning, so we have these point locations or sample data, like the equivalent. Then we have the variables, and all those will be processed within GRASS in this case. And then we will move to R for the modeling part. Okay. Okay, we will use then data obtained from GBIF for the mosquito and data provided by Mundialis for LSD. It's a reconstructed data set daily at 1 kilometer spatial resolution, and it's already converted to Celsius degrees. Okay, so you can get all this data from the links here or in the README of the report that I pasted on the Mattermost channel. Basically, it is a ready-to-use location, GRASS location, and within it, there's a map set. We will just see what all these terms mean. And then download the mosquito occurrences also as a geo package. And you can follow the GRASS script and the R script as well. I will be just showing it within the presentation. In the end, what you should get on your computer to then reproduce this is a folder called GRASS Data or GRASS Data Open to Your Hub, as you like to name it. And within that folder, you download and unzip this location here. Okay, and then you will have this tree structure, this directory tree structure. Okay. Okay, so when you try to open GRASS, you are presented with this screen. See, and basically this will completely disappear in GRASS 8, but we are not yet there. We are almost about to release GRASS 8, but not yet. But basically, it asks for 3 directories, let's say. The GRASS data, within the GRASS data allocation, which is like the project, and within that project, like a sub-project or what we call map set. Okay, and then you start. Yeah? Now, what is all that? This GRASS data is just a directory. It can be on your local machine, it can be on a cluster, it can be in the cloud, okay? Within that directory, you will have all your GRASS projects, and we call them locations. It's like some kind of legacy term, okay? But basically, each location is defined by its coordinate systems, okay? So everything within a location will necessarily have the same coordinate reference systems. And with that, you forget about overlapping problems, as it is so usual in GIS environments, okay? And then map sets are just like sub-projects within this location, and you can organize them like for different areas or different topics or whatever you like, basically. So when you start GRASS, you are asked for these 3 clicks. Basically, you are being asked for a path, okay? Which is composed of this GRASS data, then the location, and then the map set, okay? GRASS needs that to start. You can follow this link or in the extra slides at the last part of the presentation for more details. Okay, so we start GRASS. Now we're ready to start. What we do, so I'm using basically the command line here. Okay, let me show you for a moment. But all this can also be done with the graphical user interface. Okay, it takes a while now to load because I'm presenting and streaming. Yeah. So like this. I already have the data import. I just want to show you this. So I'll go here to the data. Okay, vector. And basically, I just display the occurrences. Okay, I can add Italy borders. So it's just simple GIS stuff. Good. Okay, back to the presentation. Oops. Well, so I import the data. Then I will just display it as I did before with clicking. I can also do with commands. So I list the raster data because I want a background for my points. I set the color palette to Celsius to one of those maps. And then I open a monitor, display the raster and display the vector. And what I get is something like this. You could also, here I just provide you with the points, but there's also an add-on in GRASS called v in PyGBIF, and you can use that to query all the GBIF database, okay, and download the data you want. Good. So then the next step, we have the presence records. The next step is to get background points. Why we call them background points? We will be using maxent next, which assumes that a species distribution, that the species uses some of the available environment, and it uses only presence records. And then it just compare the environment in the presence records with the background information. You are not sure if those are absences. That's why it assumes it's just the available environment, okay? So if we then want to use a different model, for example, maybe random forest or GLM or something like that, we would need to be sure to provide absences and not background points, okay? Okay, but here what we do first is just create a buffer around the presence points, okay, because we don't want the background point in the same place. We set a mask, okay, we create a mask from one of the rasters available. And we convert that raster mask into a vector mask, and then we do just an overlay to then create the random points only within the limits of Italy, but discarding the holes that will represent the presence points. Okay. For more info about computational region and masks in GRASS, please see the extra slides in these links so you can better understand what I'm saying. Oops. Okay, wait. Yeah, here. So basically what we have now is this. The presence points are the rounds and the crosses are the background points. We created 1,000, okay? So now we have all the points part ready. Now we move to the time series part, okay? And we will be creating some aggregates from this 5 years of daily data, but aggregates that we assume that will have some meaning for the mosquitoes, okay? According to the biology of the mosquitoes, okay? So as I was showing at the in this plot with the framework, we first create the table in which we will be registering the maps, okay? So we say it's a raster time series with absolute time. We put a name to it. It will be LST daily, a title and a description. Okay, so we have daily LST, average daily LST in Celsius from 2014 to 2018. We list all the maps that we have in our map set. We create a file with 1 map per new line. And we use this file to assign timestamps to these maps and register them within this table that we created at the beginning with tcreate, okay? And here we say this is interval data because each map represents 1 day. We pass, of course, the table that we created here, the file with the list of maps, and we say, okay, it starts on January 1st from 2014, sorry, and the increment is 1 day. So according to the list of maps, it will understand, let's say, which date it has to add to each map, okay? And then we can ask info to this time series. And basically what we get after running all those commands is an output like this, in which we can see that the start time is January 1st and the end time, since this is an open interval on the right, the end time is January 1st, 2019. The granularity, as we saw before, is 1 day, and the temporal type is interval. And then we get here, total number of registered maps is 1,826. Then we get the resolution and we get general statistics for the whole time series, okay? Now, once we have registered this time series, which has 1,800 maps or more, we will be aggregating this data. So the first thing we will do is to create long-term monthly average, minimum, and maximum LST. And for this, I just wrote here a for cycle to avoid, this is bash, to avoid having to type for the all months. And if someone is using Windows, here's how it looks in Windows too. And basically we use the GRASS series, which aggregates an input time series into 1 map with a method, okay? So in this case, we say, okay, we want the average of this time series, but not the whole time series. Just pick the months that start with I. In this case, since this is a cycle, It will be 0, 1 first, then 0, 2, and so on and so forth. And the same for minimum and maximum. And basically, the output for this will be the average LST for the whole maps corresponding to January, February, March, and so on. And the same for minimum and maximum, okay? We will use these maps as the input to create bioclimatic variables the same way as WordClean does, okay? So for this, there's an add-on that we can install with G extension called r.bioclim. And basically, it needs the list of maps that we created before. Okay, so for the average, it will need all this list of maps. For the minimum, the same. Okay. And basically here, I can just paste the output of one command within the other command. And that's what I do here. So I ask the list of raster maps with this pattern and separated by comma. And the output of this will be what this parameter gets, basically, what tmin gets, okay? So since I am only providing temperature maps and not rainfall, I won't be able to estimate the 19 bioclimatic traditional variables, but only those based on temperature. Okay, so I will get from bio 1 to bio 11, more or less. And we can display one of them. Okay, this is the annual mean temperature. You can see the manual of BioClim for more details. Then we will use a different raster module, TRAST module, which is TRAST aggregate. This module allows us to aggregate with granularity. Okay, so we will again input the whole time series, but what we want is the slope of the spring warming. Okay, so we will only choose the months of February, March, and April, and that's what we do with this where clause here. And we say we want that per year, okay? Granularity 1 year. So we will get 5 maps with the slope of the spring warming. We could use those then in our modeling, or we could just estimate an average for that, okay? And that's what we do here with, average, median, or some statistics of that. And that's what we do here with TRAST series, okay? So we input the output of this one, of TRAST aggregate, and we get a map with the average slope of spring warming. Oops. Okay. And then we do the same for the other part, the cooling in autumn. Okay, so it will be basically similar, but just choosing different maps. Other thing that we can do, and now we show a different module, is, for example, count the number of days that has an average LST between 20 and 30, because that is like a kind of a very good range of temperatures for mosquitoes. Bear in mind, though, that this is LST, not air temperature, okay? So maybe in a real setting, we would need to adjust these thresholds, okay? But to exemplify how TRAST algebra works, we just write this expression. So this will be the output time series, and we write an if clause. So if the input time series, like every pixel of every map of this input time series, is higher than 20 or lower than 30, just put 1 in that pixel, otherwise write null, okay? So basically we will get a new time series with ones and nulls. And then we aggregate that time series, okay, with a count method and a granularity 1 year. So we will get a new time series of 5 maps, and each map will have per pixel the number of the number of times that this condition is met. Okay, we could then aggregate that to an average, for example, to get only 1 map. And this is what we get as an average. Okay, so the average number of days with temperatures between 20 and 30. And yet more complicated example, this is the last one, I promise. We want to know, for example, the number of consecutive days that meet a condition. So for mosquitoes, for example, the minimum temperature is a limiting factor, okay, because the eggs die, basically. So let's say, for example, some authors say that -2 is this lower threshold for survival of eggs. So let's try to count the number of consecutive days, because this is important. Maybe if only 1 day is -2, it doesn't matter, but if there are many consecutive days with this temperature, then eggs die. So how do we do that with T-GRASS? Okay, we need to count that per year. Okay, that's our basis. We could do it also per month. Let's say we do it per year. We need to create a time series with yearly granularity. Okay, with any method, it doesn't matter. We choose count. So we will get 5 maps, and the values will be 30. 370, 65 or 366, okay? But to add to those will be like a bit odd, so we convert that to zeros. So basically the second command does what we call a mask, an annual mask, in which maps values are 0. Why? Because then we will be adding to those maps in a pixel based. So, and here's the core of this operation. We used algebra again. And here's the output time series, okay, lower -2 consecutive days. And we say, okay, to the annual mask, add where the maps are contained to the left. So add to the left, meaning add here in maps of this time series, if the LST of the current map is lower than -2, and the LST of the previous map of the current one is -2, or if the current map -2 and the next one is -2. So like that, we are adding 1 to the count of the base map of the annual mask, okay? And importantly, most of these temporal modules are parallelized, so you can set the number of processes you want to use to run in parallel, okay? Because otherwise, to do all these evaluations takes a while. But here, for example, I use 7, and it's just like okay, and then we can also aggregate the result of the algebra with the median, for example, okay? And we get the median number of consecutive days with temperatures below -2. Of course, in the mountains, it's a lot, and in the rest is almost nothing, okay? Okay, now we have all these maps that we have generated. Now, how do we connect with R? As I mentioned earlier, there's this package called R GRASS 7 by Roger Vivant mostly. So here a note, volunteers needed to help Roger, please. He did a great job all these years, and now he is retiring, and please, we need volunteers here to help. But basically, this package has a few functions. One is the init grass.
It will be difficult to replace, I can tell you.
Yes, I fully agree. This function init grass will start a grass session from R. Exec grass will allow us to run grass commands within R, sorry. And then with read vect and read rust, we basically read the maps from the grass database into our objects. And with write vect and write rust, we do the opposite. Okay, we write back into the grass database. Now, as I said before as well, there are two ways of connecting these two softwares. Oh, sorry, my belly is making noises. We can use R within a grass session, okay, like this, like in this screenshot. I was in the grass terminal, I type R, and R just starts there, and I get the R prompt, okay? So there I would just load our grass library and then use read vect or read rust to read the data and run exec grass to run the modules and so on. Or without starting grass, we just get some parameters needed to start grass, like the location, the map set, all the things that we mentioned at the beginning. We get all those parameters and we start grass from R. So without starting grass at all, okay? And here, indeed, we need this init grass. This was, according to Roger, originally intended to just apply grass functions on data outside grass database. So some people prefer to create like throwaway locations here, but I really like to use grass from R in this way, and this is what we will be using now, what I will show now. So basically, if you were running grass, you closed it, just open RStudio or R, how you like it, load the library, and then we set where the grass binaries are, which is the grass data, the location, and the map set, and we run init grass, okay? And this will give us like the data, the metadata of the grass session. So where are you running grass? Where R started grass, and the region, okay? And then a lot of information about the projection. Then we just load other libraries that we will be using: raster, simple feature, map view, and biomod. This one is the one that we will be using for the species distribution model. Okay, so to read vector data into grass, we can use either SP or SF, so spatial or simple features. I like simple features best, so we use simple features. We read the presence records, the background records, and we just display them quickly with map view, okay? And I really like map view because it's really simple to show data. Then we read the raster data. So for raster data, we currently can only use SP. So we have to specify that. Otherwise, it tries to load them as star objects, but that is not supported yet. So we specify this, and then we just run exec grass and a glist. basically trying to search for the rasters that we have created, okay? All the working or all the averages, all the median raster maps that we have created. We concatenate them in a string vector and we import them, okay? And while importing them, I just converted them to rasters. Otherwise, they are like spatial grid data frame or so, and I prefer the raster. And we display one of them, basically. Okay, so let's say the bioclim one with map view. So see, we are reading like GRASS data within R. Then we just follow some data preparation and formatting required for the species distribution modeling. Okay, we just need to specify the coordinates, the name of the species, and so on. And also we stack the raster predictors that we imported and create a biomode object with all this information. And we can also show it. Basically, what it does is estimate the univariate statistics for all the presence and background points. And if we plot it, it will show us pretty much the same that we had in GRASS, okay? It's difficult to see here, but they are plotted with different colors, with green and kind of red, dark red. Okay, but these are the commands. Then we need to set some configurations for the model that we will use. In this case, since we will use Maxent, we need to provide the path to the Maxent jar file. Okay, and some other like number of simulations and so on. And this is just a summary of this object. And then we basically run the modeling. And here we can set how many runs to evaluate and how to split data to do the cross-validation within the modeling, and which evaluation metrics we will use. This called rock here is actually the area under the rock curve. Then we get evaluations from once the model run. I don't run it live because it takes a while. That's why I chose just to put everything in the presentation. We get the evaluations from the object, the output of the model, and we can print it here. So this will be the accuracy, and this will be the area under the rock curve. Okay, not so fascinating values indeed. And we can also plot them like area under the rock curve versus accuracy. And we get like the maximum of both. We can also estimate the variable importance, similar to what Hannah showed at the beginning, but the function in this package. And we can show them for all the runs that we run, or we can estimate a mean. Okay, so this is only the head of the table, just to fit the slide. Then we can, other thing that is usually done in species distribution modeling is estimate the response curves that shows us how the relationship between the predictors and the response is fitted. And here we obtain that. So all the variables that we used, how the accent in each run fitted the relationship, okay? Some are more or less similar and others are just, we have like little or small data set. And then finally, we just project. So this was fitted, this was fitted just for the presence and the background data, okay? But now we want to project the results into the space of our predictor variables. And so we use this biomode projection. And we basically give it the raster stack with all the predictor variables. And with get predictions, we get the projections and we just display the one with the maximum area under the rock curve. Okay. And this is basically the result. So I think here, down here is Milano, so all around the cities, these mosquitoes thrive. And then optionally, we can write the raster data back to GRASS. Okay.
Where are you leaving some time for? Where are you leaving some time for questions? We are like
I'm just, yes,
7 minutes.
I'm
okay.
Almost done. Yes,
Thank
this
you.
is just to show how to write back. And a disclaimer, because this is just a toy example, but with a lot of things. But just to say that, of course, there are other models to test. You can also do hyperparameter tuning, variable selection, model selection, ensemble modeling, validation. We are not doing validation here. And of course, there's the issue that Hannah showed at the beginning, like where can we really predict with confidence? And of course, there are many other relevant packages that could be used. So this was just an example of how to connect the two softwares and so on. How to get GRASS help. If you are interested, there's a lot of other workshops and links that you can follow, references, and see you at Post4G.
Good, thanks so much. Thank you so much, Veronica. Yeah. There are a couple of questions on the Mattermost channel. Not that many, by the way. So a question is, can some of the computations be conducted in GRASS, be run in parallel or run on a cloud server?
Yes, indeed. In parallel, especially the temporal modules, most of them has this parameter and props. So you can set how many works in parallel. And one other nice thing of GRASS is this map set concept. So you could even like split your raster into pieces and put them into different map sets. And since the sessions are kind of independent, you could be running them in parallel and then just paste everything together, patch together. And the other option is now just deploy Actinia and GRASS in a cloud environment, and that would allow you to for a different setting as well.
Thank you. You mentioned GRASS version 8. Can you explain a bit more on the most exciting new features that will be added to version 8?
Yeah, there's some GRASS fun there.
Shortly.
Okay, so yes, basically we will remove the startup. This okay, let me show. The startup where you need to choose the GRASS data, the location, the map set that people usually gets. People really get scared with that, and most people cannot pass from that screen, so that will be completely removed. And if you don't have a GRASS data already in your computer, and it's the first time you open GRASS, you will be presented with this screen. So you directly enter the software and automatically you will get a GRASS data and a lat-long location with these maps inside. And this note that you can proceed like using this location or create a new location and with explanations and so on. And this is for GRASS 8. For GRASS 8 Plus, we are expecting to have, now this is like super experimental, a single window display where you can just move these panes around and get the console or get the Python terminal here and just go
looks
with
like
a single
QGIS. Looks like QGIS a lot, which is good.
Yes, yes, I really like it. So this is the output of one of the Google Summer of Code projects. Yes. And then there are like many other like code quality stuff and new add-ons and new other features, but GRASS 8 is mostly about working on the GUI.
Good. And some not easy scripting environment for GRASS. Are you working on that also, or because
yes.
you know?
Yes. So there's this new also from Google Summer of Code project that just finishes, GRASS Jupyter library now, and it allows to connect much easily to Jupyter notebooks. So you can do like this interactive GRASS scripting there.
Mm-hmm. And maybe something for RStudio also, do you think?
Hang on, Tom, we have questions on the Mattermost.
Okay, sorry.
Not yet for RStudios. We need volunteers for that. Most people use Python with GRASS.
Okay. Sorry, Edzer. Go ahead.
But there was a question from the question and answer window. Can you compare GRASS with Saga briefly?
Being a GRASS user, I do not use Saga. So that's my disclaimer.
No is a good answer, right?
But so I cannot be fair to compare because I really don't use it. What I know is that Saga has a lot of hydrology models and so on, and that's why most people doing those kind of analysis use Saga. But yeah, I wouldn't be fair to say anything because I really don't know Saga.
Good. Are there plans to make GRASS data directly available to web map servers?
I think there was. I think Marcus would know. I think Mundialis was developing something like to connect GRASS data directly, like a plugin or something directly to WebGIS.
Okay, good.
So to avoid going through all the different steps.
The one but final question, can the results obtained from Maxent models trained using biomod2 package be used in r.maxent.lambda's GRASS add-on to predict presence probability?
I think grass.maxent.lambda, there you only pass like the coefficients of the Maxent, and GRASS with those coefficients builds up the prediction. So you would be doing the same basically.
Yeah.
It's like parallel.
Yeah.
Exactly.
And a final question, GRASS versus Quantum GIS. Is it fair to consider GRASS more as a geoprocessing tool and Quantum GIS more to make layouted maps?
Yeah. Yes. Yeah. I mean, I use GRASS mostly for, let's say, processing. So most people, when you say GIS, they think it's like clicks and vector data, but GRASS, though having all these clicks and vector data modules, it is, for me, in my point of view, much stronger in raster processing and image analysis and raster time series processing. So I basically use it to do all this. And if I want to then create a map for a publication or visualize data, I use either R or QGIS, mostly
Okay.
R.
Let me thank Veronica very much for giving this very enlightening presentation on GRASS and time GRASS and integration with R.